You, in the name of God. Hello, everyone. I'm so sorry for the delay and other problem, and also thanks the manager team for their supporting. This is Dr. Najmeh Kaffash Farkhad, PhD in Immunology from Mashhad University of Medical Sciences, Mashhad, Iran. I will be talking around the article entitled Are Mesenchymal Stem Cells or MSCs able to manage cytokine storm in COVID-19 patients? A review of recent studies. This review article aims to collect available clinical data in more detail and investigate the role of MSCs in reducing the cytokine storm and improving clinical parameters of COVID-19 patients for use in future clinical studies. The next, please, slide. Thank you. As you know, the COVID-19 is the biggest challenge in the medical world nowadays, and all scientists around the world are trying to find a way to tackle this problem. The prevalence of new coronavirus or SARS-CoV-2 or acute respiratory syndrome coronavirus 2 causing lung infection started in December 2019 in Wuhan, China, and the virus spread rapidly around the world. SARS-CoV-2 is an envelope virus having a positive sense single-stranded RNA as a member of the lineage coronavirus family. And as you see in this picture, it has four primary genes encode the structure of proteins, including nucleocapsid protein or M protein, the spike protein, envelope protein, and the membrane glycoprotein or M protein. And as you see, S protein has two subunits, S1 and S2. And the main task is to mediate the entry of infecting viral particulates in the host cell. It has been proven that the virus usually passes through the mucous membrane, especially the mucous membrane of the larynx and nose in the respiratory tract, and reaches the lungs. Then it enters the peripheral blood from the lungs and causes viremia. Afterward, the virus attacks the organs that have the angiotensin converting enzyme 2 receptor or AC2 receptor. And since the AC2 receptor exists on various cells, including epithelial lung cell, kidney, heart, and liver parenchymal cells, it can cause multi-organ failure in severe COVID-19 patients. The next slide, please. Thank you. The COVID-19 has a wide range of clinical symptoms, including cough, fever, lymphopenia, sore throat, fatigue, headache, and sense of taste or smells loss. And it has severe complications like multi-organ failure and acute respiratory distress syndrome or ARDS in critical patients. Also, this infection is correlated with a destructive inflammatory response, including releasing many pro-inflammatory cytokines called cytokine storm. In summary, and as shown in this schematic diagram, the cytokine storm occurs when the coronavirus enters the body's immune system through ACE2 receptor available on the surface of immune system cells, and in response, NF-kappa pathway is activated, leads to overproduction of inflammatory cytokines and factors called cytokine storm. Subsequently, the cytokine storm affects the immune system and the cycle repeats itself again and again if not inhibited. The next, please. Thank you. So the main question is that what's the cytokine storm and how it happens? Cytokine storm is an inflammatory phenomenon which is marked by the systemic release of large number of inflammatory cytokines, including interleukin-6, interferon gamma, tumor necrosis factor alpha, and etc. The release of a large number of free radicals from immune cells is induced by the cytokine, leads to cytokine storm. And as you see in this schematic diagram, in the field of COVID-19 disease, some changes occur in the innate and adaptive immune system, which leads to cytokine storm formation, including increasing in the number and activity of monocytes and macrophages, especially CD14 positive monocytes or M1, with high inflammatory gene expression as a main antigen presenting cells, which leads to overactivation of TCD4 and TCD8 positive lymphocytes. Subsequently, overactivated TCD8 positive lymphocytes characterized by overexpression of inflammatory markers like CD38, PD-1 program cell death, on their surface secret large amounts of inflammatory interferon gamma cytokine. TCD4 positive lymphocytes also became overactive, especially those inflammatory subsets, including TLP1 and TLP17, which leads to increased secretion of inflammatory cytokines like interleukin 6, interferon gamma, and TNF alpha. Furthermore, activated TLP17 cells cause massive degranulation. However, in comparison, the number of anti-inflammatory lymphocyte subsets, including T helper 2 and Treg, and subsequently secretion of anti-inflammatory cytokines like interleukin 4, interleukin 10, and TGF beta is reduced. Also, downregulation B cell activation pathway in the severe patients indicating dysfunction of these cells in COVID-19 disease. On the other hand, and as you know, you can see in the uh, left part of the picture, eosinophils number decrease in COVID-19 patients. As you know, eosinophil can induce CD8 positive T cell proliferation and produce nitric oxide and thus play an influential role in antiviral defense. Natural killer cell dysfunction and decreased their number is another common alteration in the innate immune system following SARS-CoV-2 attack. By secreting large amount of cytokines like interferon gamma, TNF alpha, interleukin 18, and chemokines, these cells play a crucial role in viral clearance. Also, by releasing cytotoxic molecules like perforin and granzyme, they can lyse viral infected cells through apoptosis induction. In addition, excessive increase in the number of neutrophils in the blood or neutrophilia, change in their function and phenotype or other alters in the innate immune system of severe COVID-19 patients. Also, the net with this phenomenon and the formation of neutrophil extracellular trap in nuclei is another mechanism involved in eliminating pathogens by neutrophils, which is more activated in COVID-19 patients. Taken together, all these inflammatory processes cause cytokine storm in COVID-19 patients. The next slide, please. Next, please. Thank you. Uh, and the important cytokines involved in cytokine storm are interleukin 6, TNF alpha interferon family, and interleukin 1 family. And as you know, interleukin 6 is produced by approximately all stromal cells and competent immune cells, including macrophage, mast cell, T lymphocyte, dendritic cells, monocyte, B lymphocyte, and some other non-lymphocytic cells such as tumor cells, fibroblasts, and endothelial cells. It is produced in infection damage and transmit an alerting sign to the entire body. Interleukin 6 is responsible for activating TLP17 cells in the interaction between T cell and dendritic cells. It is recognized that interleukin 6 and D dimer levels dramatically increase in severe COVID-19 patients, and so they can be good diagnostic predictors for finding the severity of the disease. Many laboratory tests have indicated that interleukin 6 boosts macrophage activation syndrome, triggers mass manufactures of pro-inflammatory cytokines, and inducing fibroblasts and neutrophils migration into the alveolar epithelium. This results in an enlarged deposition of fibrin and collagen, leading to harm to underlying lung tissue. And TNF alpha as a dominant pro-inflammatory cytokine is applied pleiotropic impacts on distinct cell types and critically is involved in viral disease associated with the pathogenesis of chronic inflammation. It released by activated monocytes. It has been proven that TNF alpha triggers macrophage-induced angiogenesis, which is a virtual events through inflammation, tumor growth, and wound repair. It is accepted that TNF alpha and other cytokines in the TNF alpha TNF receptor superfamily are potent inducer of NF capability leading to multiple expression pro-inflammatory genes. Also obtained data from several studies show that anti-TNF alpha therapy reduced morbidity and mortality in COVID-19 patients. Interferon families are another important cytokines involved in cytokine storm. This family is a set of cytokines secreted by cells over specific situations like exposure to viruses, polypeptides, or double-stranded RNA, and have crucial immunomodulatory, antiviral, antitumor, and antiproliferative properties. In mammals, the system is the first protective barrier against viral infection. Type 1 interferons, including interferon alpha and beta, uh, as an essential part of the intrinsic antiviral innate immune system, are the fastest and most effective antiviral response as soon as the virus enters the body. Viral infections, including SARS-CoV-2, can suppress the production of interferon type 1 and 3, but induce the production of interleukin 6 and TNF. Interferon type 1 might remarkably reduce viral replication. Counter studies show that interferon disorder is an important marker to discover COVID-19 pathogenesis. And efficient interferon stimulation or prophylactic direction of interferons at the earliest stage before severe COVID-19 may bring about an autonomous antiviral state, limit the virus infection, and inhibit the COVID-19 progression. And the interleukin-1 family, which is consists of 11 members, is another important cytokine family. Some more important of them are interleukin-1 alpha, interleukin-1 beta, interleukin receptor antagonist, interleukin-18, and interleukin-33. During the cytokine storm, interleukin-1 beta, interleukin-80, and interleukin-133 are the three most essential cytokines from this family. Thank you. The next slide, please. Thanks. In this slide, you can see current treatments and limitations for COVID-19 disease. As you know, so far, many treatment strategies for this disease have been proposed, including hydroxychloroquine and combinations, neutralizing antibodies, repurposing various antiviral treatments, and passive antibody transfer from convalescent patients' sera. There are still controversies about these treatments, and they have a complementary role. Besides, they seem insufficient due to the rapid progression of the disease and the increase in the number of patients and certain limitation of any treatment methods. For example, using high doses of steroid is not only useful in treating COVID-19 patients, but can also cause severe side effects like the increased risk of bacterial infection or diabetes. On the other hand, considering differences in the severity of the disease and individual discrepancies in the incidence of immune responses to some existing treatments, the need to find new treatment strategies is felt. In addition to inhibiting virus replication, perhaps one of the strategies to improve critically COVID-19 patients with severe lung involvement is to prevent cytokine storm formation. Some new therapeutic strategies like cell therapy may be a good option. Mesenchymal stem cell or MSCs and their derivatives may be able to prevent or mitigate the cytokine storms through their immunomodulatory capacity and lack of expression of ACE2. The next slide, please. Thanks. What's the mesenchymal stem cell therapy? Mesenchymal stem cells are adult multipotent cells with self-renewal potential and multilineage differentiation ability into a specialized cells. Their safety and potency have been recorded in several clinical trials, especially in diabetes, immunomediated inflammatory disease like graft versus host disease, and systemic lupus erythematosus, amyotrophic lateral sclerosis, and also in COVID-19. MSC therapy can theoretically suppress the overactive immune system and by improving the microenvironment assist endogenous restoration. MSCs exert their effects mainly by immunomodulatory and differentiation ability. The next slide, please. Next. Thank you. What's the effect of MSS and ARDS? ARDS or acute respiratory distress syndrome is the main typical intricacy of COVID-19 disease causing by various events, including the renin-angiotensin system dysregulation, cytokine storm overactivation of neutrophils, and elevated coagulation. The ARDS usually involves a general lung injury that is associated with characteristics such as pulmonary edema and damage to the endothelium of the lungs. Unfortunately, the mortality rate of this complication is relatively high. Many studies show that MSCs are useful in the treatment of ARDS. After intravenously reaching the body, some of the MSC accumulate in the lung and potentially prevent pulmonary fibrosis, protect lung pneumosis, improve pulmonary microenvironment, and enhance lung function. Besides, because of ATP levels, reduction in injured alveolar epithelial cells as another mechanism, MSC by transporting mitochondria to these locations and refilling depleted ATPs improve their function status. And the next slide, please. Next. Please, next. Thank you so much. And how mesenchymal stem cells can manage cytokine storm in COVID-19 patients? In this schematic diagram, you can see the underlying mechanism of MSCs and cytokine storm management in COVID-19 patients. The immunomodulatory effect of MSCs are triggered by the activation of toll-like receptor in MSC is stimulated by the pathogen-associated molecules such as LPS or double-stranded RNA from the virus. In response to the virus entering the body, mesenchymal stem cells secrete several soluble factors, as you see, including nitric oxide, inulamin 2 deoxygenase, prostaglandin E2, and transforming growth factor beta or TGF beta, which affect the immune system in two ways. On the other hand, the soluble factor inhibit activation and proliferation of TLP1 and TLP17 cells, which subsequently leads to a decrease in inflammatory cytokines, interferon gamma, and interleukin 17. Besides, with the help of these soluble factors, MSC exert immunosuppressive effects on dendritic cell as a central antigen presenting cell by inhibiting dendritic cell activation, decreasing interleukin 12 production, decreasing endocytosis, and inhibiting dendritic formation cells from monocyte and cell maturation arrest. This results is an increase in interleukin 10 and a decrease in TNF alpha levels. Also, MSC can suppress NK cells in two ways: direct contact with them and indirect interaction by releasing mentioned soluble factors. It has also been proven that during the progression of COVID-19 disease, CD840 T cells, CD8 positive T cells become overactive and attack lung parenchyma to remove the virus. Mesenchymal stem cells prevent serious lung damage by inhibiting disactivated lymphocytes. Moreover, in COVID-19 patients, MSC prevent pulmonary fibrosis by decreasing the profibrotic factors levels and improving the lung microenvironment. Also, in another mechanism, MSC causes remodeling and shift the T cell subset to regulatory cells, and subsequently they increase the level of anti-inflammatory cytokine interleukin 10, which plays a crucial role in regulating immune response. And thank you. The next slide, please. Thank you. In this slide and the next few slides, you can see the result of several clinical trials in which MSCs have been used to treat COVID-19 patients. So far, more than 19 clinical trials in the field of stem cell therapy have been registered for COVID-19 patients in clinicaltrial.gov sites. Most of them are in the process of being completed, and only if you have reached an end and published the result. The results are promising and no serious side effects have been reported during the patient's treatment period. For example, as you see here in a case report study by Tao et al., inject 1.5 million umbilical cord blood mesenchymal stem cells 5 times in a patient's 10-day follow-up period could improve the patient's respiratory and renal function. Additionally, no serious side effects were observed. The next slide. And, okay, and the next, please. 12th slide, please. Thank you. Also, similar results have been reported in other studies with larger population. For example, in the study of Hashemian et al. On 11 COVID-19 patients, injection of 200 million stem cells for 3 times significantly improved oxygen saturation and lung tissue regeneration. The cells also successfully reduced the cytokine storm, which was reported by reducing the serum level of inflammatory cytokines, interleukin 8, TNF alpha, interferon gamma, and interleukin 6. In the next few slides, similar results of reducing cytokine storm and improving the condition of patient after cell injection can be seen. Please, next slide. 2:15. Next slide, please. Thanks. And the next. Thank you. And in this context, our research team is also conducting a cell therapy project for COVID-19 patients. You can see me in this picture. And in summarize, we also use umbilical cord mesenchymal stem cells injection in 10 COVID-19 patients compared to 10 COVID-19 patients in the control group who receive only standard care without cells. The findings of this study were also promising, and the results will be published soon. Thank you. The next slide, please. Okay. As mentioned earlier, the coronavirus can reach target organs through the ACE2 receptor at the cell surface and exert its destructive effects. And because the ACE2 receptor is present on almost most cells in the body, multi-organ failure occurs in the later stage of the disease. Some of the most important target organs that are affected by the coronavirus are lung, kidney, and liver. Evidence from studies shows that MSCs can have beneficial effects of any of these organs, including the next slide, please. Next, please. Okay. The effects of MSCs on lungs. After intravenous injection, MSC by homing in the lungs boosts their function via multiple mechanisms, including avoiding pulmonary fibrosis, preserving lung pneumosis, enhancing the pulmonary microenvironment, protect endothelial cells from inflammation and oxidative stress, raising autophagy through the phosphoinositide kinase protein B pathway that may defend alveolar cells and secrete high levels of growth factors with critical role in tissue repair. Okay, the next, please. Thank you. MSC also can affect kidneys. Indeed, kidneys are the second most affected organ in COVID-19 disease behind the lung. Many studies have been reported elevated serum creatinine and urea in COVID-19 patients. MSC defend against acute renal injury and enhanced renal restoration in acute and chronic kidney injury models. Intravenous injection MSC have moved to tubular glomerular interstitium and peritubular capillaries and exert the immunomodulators effects in this device. In many studies, the amount of serum creatinine, blood urea, nitrogen, and renal parameters after injection of MSC return to the normal levels, which indicates the beneficial therapeutic effects of these cells in the regeneration of kidney tissue and reduce inflammation in it. Okay, and the next slide, please. Liver, okay. MSC have also beneficial effects on liver. Liver dysfunction is common in COVID-19 patients, approximately between 20 to 50% of patients. However, there's still controversy about the correlation of these findings and the disease prognosis or progression. It has been documented that SARS-CoV-2 can affect liver cells by causing elevated levels of aminotransferase enzymes and liver impairment. Two hypotheses have been proposed in different studies in liver dysfunction and infection with coronavirus family viruses, including systemic inflammation and direct liver attack by the virus. In this regard, MSCs are useful in improving liver problems via inhibiting growth formation and improving liver injury and decline liver inflammation and regeneration hepatic tissue. And the next slide, please. And in summary, as you know, the pandemic of COVID-19 infection caused by the highly pathogenic virus SARS-CoV-2 has prompted an urgent need for novel therapies. It is proved that SARS-CoV-2 in some patients leads to induction of excessive and prolonged inflammatory responses, leading to hyperinflammation called cytokine storm. In COVID-19 patients, especially those with severe status, cytokine storm inhibition by immunosuppressive is urgent. Preclinical and preliminary clinical data suggest that MSCs, through their anti-inflammatory and immunomodulatory actions, can significantly inhibit this process, heal tissue therapy, enhancing recovery. Of course, further clinical investigation with a larger sample size are still needed in this area. And the next slide. Okay, and at the end of my talking, I must appreciate all the attendants who listened to me, and I wish God peace and patience for who died of coronavirus disease and peace for their families. I hope there will be peace in the whole world, and the countries will be friends and brothers together. And my message as an Iranian researcher is peace, love, and respect for everyone. And I also thank all the facilities who give me this opportunity to talk and present. Absolutely, my colleague, especially my best professor, Dr. Tawakkol Afshari, and my dear husband for their supporting. I'm hopefully one day we will be together in other conferences and webinars and have a great time and goodbye.